Dr. Vivek Das will be enlightening us with his knowledge on role of bioinformatics in the present era of high-throughput biology, digital health, and drug discovery. And let me introduce Dr. Vivek. He was born and raised in India in the city of Joy, Kolkata. Vivek Das, doctor, has been quite the wanderer, having lived in various parts of the country and the world, from having a bachelor's degree in biotechnology from Bangalore University, master's in bioinformatics from BIT Mesra Ranchi, IT programmer analyst job he did in Cognizant, Kolkata. He holds a PhD and a postdoc in cancer computational biology from IEO, Milan, and industry postdoc at Novo Nordisk, Seattle. Dr. Vivek's professional career has a global footprint. Presently, Dr. Vivek's passion brought him to Denmark, where he is a computational biology senior research scientist at Bioinformatics and Data Mining Department in Novo Nordisk. He is involved in biomarker and target discovery in chronic diseases using in silico approaches. His present work involves crunching big data from bulk, single-cell multiomics, along with phenotypic measurements that need careful integration of interdisciplinary knowledge from biology, computer science, mathematics, and statistics. He also explores the application of advanced statistical machine learning and deep learning models in high-dimensional single-cell multiomics for particular problem scopes given the data bandwidth as a part of some of his curiosity-driven research activities. And last but not the least, his hobbies include science communication, remote mentoring, blogging, traveling, functional training, hiking, and reading. And for more, you can follow him on his LinkedIn profile. I'm excited to hear your talk, sir. We all are indeed. Over to you.
Thank you. Thanks for the kind introduction. Can you hear me?
Yes, sir, we can hear you.
Yes, so I'll try to share my screen. Can you see my slide deck?
Yes, we can see a slide deck.
Okay. Thank you, everyone, and thanks for having me here for the day 2 of the Omics Research Symposium on the data and life science getting important, making impact. Today, I will speak about the role of bioinformatics in present era of high-throughput biology, digital health, and drug discovery, and what are the kind of work I've been doing at Novo Nordisk, and some of the gigs that I've been doing outside as a part of my curiosity-driven research. So before going ahead, I just want to present what's the perspective of how do people see bioinformaticians or computational biologists. And there are different aspects, a different perspective how people see us. It's sometimes very amazing, very funny, and I would encourage everyone to actually think like how bioinformatics is perceived in today's world. It's a very exciting field actually, and you must have all seen how actually everybody has explained what its role is. So I will just put some flavors of how I see it and how I've been using it. I basically am this person who is basically doing stuff with data. These data are generated in-house from different therapy areas, different biological scientists, or from public level resources. And then I use algorithms or methods that are already developed or we are developing in-house to crunch these data in a way that we can make novel discoveries out of it. That can help us to realize early drug targets or even biomarkers for onset of the disease or even disease progression. But the overall goal is to actually feed them into these 4 cornerstones of medicine, which is prediction, prevention, personalization, and participation. This is the 4 corners that I think bioinformatics is slowly emerging as a leading interdisciplinary field. Moving ahead, I would first like to give an overview of what I do as a scientist in our department, Novo Nordisk. We are a big pool of 30 plus people now across multiple sites at Novo Nordisk. I belong to the bioinformatics and data mining team, and we stand on these 4 pillars. We serve multiple therapy area R&Ds. We serve different research technologies where we provide different bioinformatics solutions. We also serve, providing solutions to understand mechanism of actions by using leveraging the potential of sequencing in clinical trials. We are trying to understand where clinical sequencing can help to get better understanding of mechanistic studies in clinical trials. So we have some new trials that we are using the role of sequencing, more precisely single-cell sequencing. And we're also, working in translational medicine. But what we do basically in all these four pillars are early drug target identification, building analytical workflows for target evaluation that can be, a very fantastic target that comes from literature from particular chronic disease areas, or data visualization, because as a part of bioinformatics, we also often try to, provide solutions where biologists can, figure out the gene names, how they are changing across disease cohorts, or single cell maps, and so on and so forth. And then we do a lot of biomarker identification work.
Dr. Vivek?
Yes.
Dr. Vivek, I'm so sorry to interrupt in between, but your voice is not that clear. Can you please check for your mic?
Sorry. I'll try to increase my volume. Can you hear me now?
Yes, I can hear you now. Okay.
Okay, I apologize for that.
No, it's
So,
perfectly fine.
yeah, great, thanks. So what we basically do is we use various public and internal omics data. Example, about transcriptomics, proteomics, metabolomics, genetics, epigenomics, or single cell omics taken all together, analyze them mostly from human and also in vitro and vivo settings to derive biological insights. I have highlighted the part human is because we follow an approach in our pipeline which is more human-centric. What that does mean is we start from human data, we derive insights, we find specific targets or biomarkers, and then we try to understand what setting of different in vivo and in vitro can help us to reach the clinical phase. So that's why we start with human data, and we have a very scalable pipeline across in a human-centric manner. Now, I will go more into the details of the talk. The outline is divided in three parts. What is bioinformatics in time of high-throughput biology and digital health? Why is bioinformatics important in drug discovery? And what is the future state and aspirations of modern medicine? Now, these are all my own personal perspectives and opinion based on my experience. And so it might not always align with everyone, but this is how I see the field and how I see the role of bioinformatics in a pharmaceutical setting and beyond. So what is bioinformatics in the time of high-throughput biology? Now, modern medicine and biology has gone a tremendous improvement and evolution. I would say it's a revolution. In current age where we have so much of high throughput, technologies where we can measure different modalities from, we can do imaging, we can understand tissue cellular composition, we can do genotyping. We can actually also leverage the powerful omics like genomics, transcriptomics, proteomics, not only from organs, but our tissues or cells, but also in vitro settings or in vivo settings. All these taken together, when we sequence them, we can actually build up a map or network at a cellular level, right? And these, when put into sequencing machines, can actually build a huge information complex of multiomics information that can help us to tease out the different molecular features, which when integrated properly, can help us to understand the disease progression or the kind of disease drivers. And what this gives us actually to help us to understand how does the central dogma of molecular biology work. I consider that as the hottest science in town, basically, because it helps us to understand the basic foundation of the entire molecular journey. And this is still the key to understand any development or disease biology. And sequencing, high throughput sequencing of different types of omics can actually do this. What it does is basically it tries to accrue all this information from different organs or tissues or cells from the human system, accrue or let's say, build or achieve the information from each type of different molecules together, stitch them with the information that we get from the clinical notes or the imaging, put them together with suitable models or methods. But in the end, it's trying to derive the relationship between the genotype and phenotype. And once we get this information, we can actually better understand a disease footprint. Now, why is bioinformatics important in drug discovery? Now, medicine over the years have been undergoing some paradigm shift. what does paradigm shift means here? We know that one size fits all theory in medicine doesn't work. Not every therapy works with for everyone. Some have benefits, some don't, some even have adverse events. So we have been seeing a change from stratified to a more precision medicine. And what precision medicine is actually ensuring is trying to say we can deliver the right intervention to the right patient at the right time. And how this can be done? This can be done with individual patient level information that can actually, take information from genomics and other omics, club them with the different information of lifestyles, their preferences, their health records, and medical history, all taken together so that we can deliver a very tailored therapeutics to each patient. And this is the dream where the medicine is actually going. And most of our industry are also trying to understand how we can actually implement this in our system when we are trying to generate new drugs or new therapeutics. And another part is over the years, we have also learned that the drug development journey is actually very, very slow and expensive. A lot of candidates fail. Not every drugs have great effects. Most are marginal. So this poses problems, right? Because it's a huge investment. So for us, it's very important now that we need to have much more clear and effective solutions. And how can we do that? With the help of sequencing or omics, and we talk about bioinformatics. Bioinformatics comes from the fact that we are able to sequence or have omics information from different layers of the molecular biology, right? And each of these plates has its own specific role to contribute to any kind of biomarker and drug target discovery in any kind of disease understanding or developmental biology understanding, as you can see from this slide, taken together with any kind of clinical information. Now, when we take together all these information, we have to be also very efficient of what models and methods we are using, right? If we are talking that we want to develop biomarkers or drug targets. Now, this whole thing is called the era of omics medicine. And I find it very enriching that over the years, due to how the field has emerged, how our scholars in the academic field has helped us, this has become now very important component also in the pharmaceutical industry. And we are now joining this journey as well. Some of the applications of omics-driven medicine where bioinformatics feed into is population genetics and genomics. Some of you might heard about, single nucleotide polymorphism or genome-wide association studies that falls into this. Then developmental biology learning or disease biology learning using different kind of multiomics of organs, tissues, and cells, longitudinal studies or cross-sectional multiomics, and then early drug discovery and therapeutics. I have highlighted some of the disease areas that we work in at Novo. Ones that are bold are the ones where I contribute and I work, and I give my scientific expertise. However, all these are great, but these are mostly done in a population of cells, population that was not having the information of cellular resolution. These lack that cell level information because we were always looking into in a more holistic manner where all the effect of these cells were actually averaged out. And human body is complex, as we know, as we are learning now of some new technologies that have emerged in the last few years, that our system has actually trillions of cells and some share commonalities and some do not. And that's when the era of the single cell technologies emerged, right? And one of the exciting example of this was this new preprint that came out from the consortium of the Human Lung Cell Atlas, where they are able to now create and map a big human cell atlas, human cell lung atlas around that maps around over 2 million cells from 444 individuals. Now, this is a massive task that has been done by some brilliant scientists from the in the field that has used bioinformatics, that have used biology, that have used interdisciplinary computer science, mathematics, and statistics and machine learning all together to understand what are the different cell types that are involved in any disease setting when we are looking into the lungs. What are the cell types that are affected by variants? What are the effects of age, smoking, and BMI, and what's not? So this is one of the biggest tasks that came out recently, and this is a wonderful example of the single cell technology. However, these kind of high throughput work is also very complex because it builds a huge information complex. It runs us into the problem of dimensionality curse, overfitting, any kind of linear, nonlinear structure, and lack of generalizability. Why? Because we are talking about matrix, like big tables, where big tables is composed of multiple dimensions. These dimensions comes from features that can be your genes, proteins, metabolites, and across your observations. Now, observations in the bulk setting is your patients, animal samples. But when you think of single cells, it's each cell coming from all these information, right? So that's a huge information complex. And this is often very challenging. And this is where bioinformatics is challenging, right? And I have discussed about it in a systematic review in 2020, where I tried to stress that how multiomics field work should be done. What are the caveats? What are the pros? What are the cons? And what do we need when we want to start on embark on this journey? And then in single cell field, also there are amazing paper out there which also speaks about what are the different challenges in these pertaining to the data science, because when we talk about these high dimensional interdisciplinary field, we know that it comes with its own caveats. And these caveats, some of them can be data sparsity, data heterogeneity, non-flexibility of the statistical frameworks, then missingness of the data, and then integration. We can tackle some of these, but they're not that easy. How we can is if we can build tools that are well benchmarked or systematically checked with other competing tools, we can solve some of these problems. But there's a lot of work that needs to be done. Not every problems have been tackled. Some of them that we have mentioned in these papers, but not everything. So there's a lot of scope of research out there, and bioinformatics has to play a lot of role in this. Now here, what I'm trying to say is, apart from the work I do as a part of a scientist for the company I work for, Novo Nordisk, I also sometimes, do work with certain students back in India, where I, put my brains in some curiosity-driven projects. And as a part of these projects, I think about problems in method development for bulk multiomics integration, as you can see here on the left. Then I'm very fascinated by the concept of single cell technologies and how high throughput biology in forms of single cell is such a big data in terms of the dimensionality. And this often begs me to question, what are the best way to actually squeeze those high dimensional data in a small latent representation so that it's human readable, human can understand and derive insights from it. And that's where the concepts of dimension reduction methods and clustering kicks in. And then when we look into these single cell, we have to have those single cell in specific clusters, and those clusters needs to have certain identity. And these are not very easy tasks, because when you're talking about labeling those clusters with certain identity where a group of cells comes together, which means that we are trying to say that that identity belongs to a specific biology, and we call them as cell type labels. And I've been thinking around those problems for quite some time, mostly on the mathematical side, like how we can get better at those cell type labelings. So these are some of the curiosity-driven problems that I've been also working as a part of my side projects. Two of them I've highlighted, and I will share a bit of those work in the next slides. One is how to better integrate multiomics data by improving feature extraction issue. And can we improve single cell annotation using a better embedding method? The first one is based on multiomics. Here in 2020, I teamed up with a few researchers back in India, where I tried to understand, can I estimate gene expression function from DNA methylation, which is an epigenomic information, and copy number variation at protein coding region. And we came up with a method of feature extraction. We thought that this is an issue of feature extraction at high dimension. We came up with a method that involves deep dataizing autoencoder and multilayer perceptron, and we took together, we were able to see if we integrate this information from DNA methylation and copy number, can we estimate mRNA? And if we can estimate this mRNA, how good is that estimate from the real data? And we used liver hepatocellular cancer carcinoma data from the Cancer Genome Atlas that had over 400 patients' data. And it was exciting to see that when we were able to actually build a model, we were able to predict the observed RNA values as good as the real ones. But that does not end the story, right? When you build a model, it's also important how good your model is when it's being tested with every other existing models that are out there. And when we did that, we benchmarked systematically a different regularization hyperparameter, how our model is working. We found that our model was actually working as one of the top models. We did in 3 different variants, and one of the variants of our models was working, came out as one of the top with the least error. So this was very encouraging for me to think how multiomics as a feature extraction issue can be actually improved. Next, I wanted to stress a bit on my single cell work that I've been doing, how I'm thinking about the field and where the problem scopes are. Same, but before that, I want to say that single cell field is massive. There's a lot of work one can do. It's beyond what we did with bulk omics or bulk multiomics. We can get information both at cell level and at gene level, which was usually we often did at gene level when we were looking into always the multiomics, right? We did, differential expression, gene set, gene regulator networks. But now we can add another layer of information. We can actually understand what is the trajectory of a disease, what is the different states of the disease, what cell states are involved, what is the composition of those, and what are the clusters that we can derive from when we are actually sequencing these cell populations. I am particularly interested in 2 problems at this moment, where I'm trying to think single cell problem scopes based on clustering and cell cluster annotation. And recently, one of the work that I have worked with 2 researchers back in India got accepted. It will be out in the next week in online, where I thought of the cell type annotation using single cell data as a problem of embedding. What I thought was, like, given we have these, huge data matrices of cell and gene, what if I think of this problem of, cell type labeling or clustering or annotation, whatever you may say, more as a problem of cell embedding and clustering. So we came up with an approach where we tried to use non-negative matrix factorization to reduce the dimension of the data of these cell costume matrices across different samples, and then try to understand if we can then additionally use recursive k-means to cluster the data, and we can actually define the cell types from limited labels that we already have from the data. And in fact, this we tested on like 6 or 7 to 8 different data sets that are publicly available thanks to the research community. And when we did any kind of accuracy and performance, we saw that our method was working very well. Interestingly, what we also did was we randomly then, missed out the real labels that were available with the data to understand how well there are cluster similarities. And we found that our methods was actually having a very high cluster similarity, as you can see from the ARI indexes for each of these data sets. Now, again, as I always say, I'm a big fan of, systematically benchmarking. I always think that why will someone use my method if I'm not actually, looking at its performance with any other methods? So I took a step back, and then I tried to understand, well, now given that we have seen that it is performing well, how does this perform with existing semi-supervised method or unsupervised method? And here there are some representative images where I was able to see that our method was actually performing very well in more than 5 data sets out of 6 when compared to another semi-supervised method that was recently published called Color, as you can see from the accuracy plots. And then when I compared the cluster similarity using against an unsupervised method called Seurat, which is very popular in the single cell field, I was able to find that at least for 50% of the data set, our method was as good as Seurat. And but it was good across almost all the data sets, apart from one from the other semi-supervised method. Then additionally, we also tested the method for intra and inter-cluster dispersion with 2 data sets that were downloaded from the European Bioinformatics Institute server, where there were real missing annotation. When I say real missing annotation, means these data sets had a cloud of cells which did not have actually any kind of cell type information. So we wanted to see whether we can redefine using our method those data sets, and can we actually get cell clusters out of it. And when we did that, we found that our method was actually outperforming the Seurat at that time. So this was very encouraging for us, that looking the cell type problem as a problem of embedding and clustering, you're able to come with a very efficient, simple, scalable method. So here I'm now progressing to the end part of my slide. The last slides are future state of aspirations of modern medicine. I see that medicine is actually going interdisciplinary in terms of cell-based intercepting. Why? Because now we have a huge explosion of single cell data, and single cell multiomics or single cell data means a lot of advanced statistical and machine learning methods, because the data dimension is huge. And this also means we are also getting better at understanding orthogonal, experimental disease models. But this is a great future for us, because now we are trying to go in a high dimensional era of medicine, where we are trying to understand the disease trajectory from healthy to different type of disease, be it neurological, cancer, or chronic. But we can actually understand one cell at a time how the disease is evolving. And we can actually detect early, which can actually understand the medicine, the molecular mechanisms, and actually, get better new drug targets. And these can all happen if we can efficiently use bioinformatics. So here I come to my last slide. I hope I was able to convey how bioinformatics is actually, helping the modern medicine and biology. All these informations that we are trying to accrue these days using molecular, spatial, molecular, histological, and anatomical. This is a huge abundance of data. These are, high dimensional data. But the overall goal is to take all these different modalities of data together, build up integrated, cell phenotypic map that can be multi-scale, multimodal, which can actually help us to understand the underlying phenotype of the cell. And when we understand the phenotype of the cell, we can better understand the states. That can be the epigenomic states, the cellular interaction, the tissue organization, and the identification of sometimes new novel cell type, which eventually can help us to understand disease mechanisms, interactions, signatures, and new biomarkers, and overall can help in designing leaner clinical trials, which can improve personalized medicine and eventually also help us building better improved drugs. So this is how I see the field of bioinformatics contributing to the modern medicine. And I hope I was able to share some of the thoughts with you all. And I thank everyone who has been instrumental in providing and guiding me with the scientific knowledge. I thank my department for supporting me and my collaborators. And I hope you guys had a good time. And this was a very nice conference. I really enjoyed everything about it. So I hope you had also some curiosity-driven questions. So feel free to ask, and I'm happy to take more questions.
Thank you so much, sir, for the presentation. It was very crisp and short and giving an overall view about how these different types of techniques are important and what bioinformatics role is in all of these studies. We have a question by Jaspreet Kaur. She's asking, what is the first step to begin with while learning bioinformatics and data mining? What basic skills are needed to start with, and please mention accessible sources.
So I would say that my field, my growth has been, I started as a biologist, and then I went on to computer science. I always see that it's important to pick up soft skills, so programming and a bit of mathematics. The way the field is progressing, one would need a bit of more understanding of mathematics. I would always say to pick up programming and mathematics or statistics a bit more if you want to venture in the modern bioinformatics. And the biology is important, but that you can learn over the line. There are some resources I can share. It's if I get an email, there are some very available free resources that is there from Swiss Institute of Bioinformatics. I keep sharing some resources on my LinkedIn profile as well. So I would be happy to share them offline to the group here.
Right, so we also have a plethora of resources, and we'll be able to share as well. Yeah, thank you so much, sir. And the next question is, what are the applications in digital health and how are they being employed in the process of drug discovery? What are your thoughts about it?
Yes. That's a great question. I didn't touch much on the digital health, but what I'm trying to portray is when we consider digital health, the digital health can be, wearables, measurements from the wearables, blood pressures, right? And these can be done from your smartwatches. And these are often your snapshot, and they are these days taken also into account when we are talking to your doctors. Now, how it will be used in a day-to-day, it's still an open research problem. FDA has not yet approved everything. We don't know how it is going to be approved, but having a blood pressure, ECG, EKG from your wearables, that's already a great data point for you to improve your lifestyle. So you can always improve your lifestyle on that front. In bioinformatics, it's still very early, but there are some trials going on. So we will learn more about it in future. At this moment, I would just say that in measuring blood pressure, it is some observational way of doing it. So yeah, that's the way I would see it.
Right. So, our next question is, can you brief on the translational potential of single cell omics and how characterizing the heterogeneity of cells will help in targeting the tumor as a whole?
Well, that's a great question. So when we talk about trials, it has to be so single cell as a diagnostic is still very early. There are some trials going on, but what we are doing is we're trying to understand, can single cell help us to understand the mechanism of action of certain drugs? So there, single cell is used quite being used right now, where we are trying to understand the role of different, semaglutides and all those things, mostly in the chronic setting. What cell picks up because the effect size is pretty low, right? That's where the single cell is actually helping a lot. Now, cell type problem is actually cell type heterogeneity in terms of tumor is a very interesting problem because it's an uncontrolled growth of the proliferation of the stuff. I think there are some very interesting paper that came out recently in drug tolerance. I think we should check on that. I don't remember the name of the author right now, but what they're trying to say is your single cell is actually trying to tell you when you're giving in tumor patients, particular solid tumor, a particular drug, how the drug is responding and what cells are responding and what cells are not responding. I think this is one of the fascinating things to understand whether the drug is actually being treated by some small cells that are responding to it, and they are actually reversing the disease. So this is how these things can help. But these are still very open research scope problem. They are not entirely solved because, as I said, the data is very, very complex and highly abundant. So it's not something that it's already solved. We are still trying to tackle these kind of massive problems as an open research problem. But there are, leaps and bounds of work that are already going on.